All right, guys, I'm back. Um, we were talking about glycolysis and how glycolysis illustrates the way that cells use biochemical pathways in order to um, push the reactions forward. So push the biochemical pathway forward. Okay, and we said that there's often some key steps that that drive the reaction, and the rest is just these concentration effects. Okay, so I'm going to go through glycolysis, which is something that you should know. So if you're not very familiar with glycolysis, you should go back to your intro bio book and review glycolysis. It's a very important basic pathway that anybody getting a master's in biology should know intimately. Okay, so glycolysis, as the name implies, starts with glucose, and it uses ATP in order to destabilize the glucose and split it into two 3-carbon sugars that are energized with these phosphate groups from the ATP. And then um, the um, the G3P, which is one of those split products, is going to be oxidized and produce NADH, and it's going to be used to phosphorylate um, ADP and produce ATP. So we get a lot of energy out of glycolysis, um, and glucose is a relatively stable molecule, and so you have glucose, which is um, going to be phosphorylated by ATP to make glucose 6-phosphate, and then you have an isomerization reaction that produces fructose 6-phosphate. Glucose is a 6-membered ring, fructose is a 5-membered ring with that 6 carbon sticking out as a branch. Um, and then you have another molecule of ATP that adds a second phosphate. So this is a pretty unstable molecule at this point, and it's going to split. And when it splits, it forms one molecule of dihydroxyacetone phosphate, or DHAP, and one molecule of glyceraldehyde 3-phosphate. These are isomers of each other, but only G3P is capable of moving through the glycolytic pathway. So dihydroxyacetone phosphate has to isomerize into G3P in order to continue with glycolysis and get any of the energy out of it. So if you look at the, the standard free energy change of dihydroxyacetone phosphate isomerizing into glyceraldehyde 3-phosphate, it's actually slightly positive. This is, at standard conditions, a slightly non-spontaneous reaction. But in the cell, we don't have standard conditions. And if you think about this, G3P is being used, right? G3P continues down this biological pathway. So as soon as G3P is formed, it's going to be used as the reactant in this oxidation reaction. So G3P is going to become 1,3-bisphosphoglycerate, and then you don't have G3P anymore because it turned into 1,3-bisphosphoglycerate in the next chemical reaction. On the other hand, you have glucose that is continuously being split into DHAP and glyceraldehyde 3-phosphate. So your concentration of DHAP is continuously increasing as glucose is split, while the concentration of G3P is continuously decreasing as it moves down the glycolytic pathway. So instead of this being a non-spontaneous reaction, it becomes spontaneous because of the concentration effects of DHAP concentration increasing while G3P concentration decreases. Okay, so um, so you can see how the concentration effects of using ATP to drive these reactions, so that we're constantly splitting fructose 1,6-bisphosphate, will allow these reactions that at standard conditions are non-spontaneous to become spontaneous in the cell. Now, sometimes that can't happen. So if we go back up here and we look at this reaction, glucose is never going to split into dihydroxyacetone phosphate and G3P or into just 3-carbon sugars on its own. You have to energize it with the ATP. And so many biological reactions are like this. If you're thinking about building proteins, or if you're thinking about building nucleic acids, or if you want to build uh, complex carbohydrates like starches, um, these are processes that take a lot of energy. Okay, biosynthesis requires energy, and so if the the delta G is so high that just increasing the concentration of the reactants or decreasing the concentration of the products, it's not going to be enough to push it in the forward direction. And when this is the case, you have to couple reactions together. So when you couple reactions together, you have your desired reaction. In this case, we want to add A and B to make AB, okay? So we have A and we have B, and we want to stick them together. But that has a large positive delta G. So we can't just throw some A and throw some B into a beaker and shake it up and have A become B in any quantifiable amounts. Okay, so in any useful amounts, right? You'll get a little bit of AB forming, but not much. So what we need to do is we need to activate one of these molecules so that we actually have a spontaneous reaction. Okay, so oftentimes we say we want to take A and add it to B to make AB. And to do that, we're going to hydrolyze ATP, right? Hydrolyzing ATP means you chop off a phosphate and you make ADP, an inorganic phosphate. And we speak of this as these are two separate events. But actually, they're not two separate events. These events combine together so that you're not adding A to B, you're adding A to an activated B phosphate. And that reaction is going to be spontaneous. So where A plus B going to AB is not spontaneous, A being added to an activated B phosphate is spontaneous. So if you look here, this is what is actually happening. We have a B, we have our B molecule, and we're going to take ATP and we're going to hydrolyze it. And in the process of hydrolysis of ATP, we're going to take the phosphate that was removed from ATP and we're going to stick it onto the B molecule. So now we don't have B, we have B phosphate. And phosphate is a very energy-rich functional group. It's got a lot of negative charge. It destabilizes molecules. So now we're not adding A to B, we're adding A to B phosphate. And while A plus B to AB is non-spontaneous, A plus B phosphate is spontaneous. Okay? Just like B plus ATP is also spontaneous. But the end result is the same. Okay? The end result is you end up with AB and you end up with ADP and inorganic phosphate. Because when you add A to B phosphate, you're removing the phosphate from the activated B. All right, I need to stop now because my baby is crying.